Hi, I'm Professor Matthew Schmidt, and this is Genetics. In today's session, we're going to begin speaking about the eukaryotic chromosome. Now, we've made reference to chromosomes in the past in a variety of ways, but I suspect that I said something like, in essence, a chromosome is just one big long DNA molecule, especially if we're talking about eukaryotic chromosomes. Well, in essence, that's true, but there's more to the story, and you see a beautiful picture of a eukaryotic chromosome here. But the way we see it this way, where it usually takes on this appearance looking like an X, when that's going on, there's a lot more than just a big long DNA molecule there. So we need to talk in this session about packaging of DNA and how that gives rise to chromosome structure in eukaryotes. Let's get some perspective first. So just for the record, in prokaryotes and bacteria, the chromosome consists of one circular double-stranded DNA molecule. Historically, and even sometimes today, it's been described as naked DNA, the implication being that it's just sitting there and there's really nothing much associated with it in a stable way. The more we learn about it, we realize that the chromosome is in a region, it's not membrane bound, but it's called the nucleoid region, and there are various proteins and RNAs involved with gene regulation and expression that are sort of in the general vicinity, and they are complexed with the DNA, albeit not in quite as orderly and organized a way as they are in eukaryotes. In viruses, again, just for the record, they do have, some people are a little bit hesitant to call them chromosomes, but they certainly have DNA or RNA, and it can be double-stranded or single-stranded. It can be circular or linear. So viral chromosomes or viral genomes are extremely varied. There's really no general rule you can make about them. But we want to focus on eukaryotes. So in eukaryotes, there are a diploid set, meaning the chromosomes are always in pairs. The actual number is not that relevant. It can be anywhere from 2 to in the hundreds. But they're in pairs and they're linear. And if we wanted to look at, say, a human individual's chromosomes, we literally can do that. An individual's complete set is referred to as a karyotype. And the arrangement is species specific. So what I mean by that, so this term karyotype is important. Karyon refers to the nucleus, right, as in eukaryon. The arrangement being species specific, what I mean is, just take a horse, because I thought of a horse. A horse has 64 chromosomes arranged in pairs, whereas humans have 46. And you'd have to be an expert to be able to do this, but for example, when they stain the chromosomes with different things, there are banding patterns that are observed, and they would be different in horse chromosome 1 than human chromosome 1. So you can make certain generalizations, but the exact karyotype is going to be different depending on whether you're looking at a human, a horse, a tree, whatever it is that's eukaryotic. Well, one of the sort of conundrums or questions that's often asked is, yes, it is true that a eukaryotic chromosome is one long, linear, double-stranded DNA molecule. But it turns out that in order for it to really fit inside the cell, it's also complexed with various proteins. And of course, it depends on the chromosome and the cell size. But the estimate is that if you took all the DNA and unwound it and just extended it out, that would be about 2 meters long, which is taller than me and probably most of you. Obviously, you have to explain how that could fit into the nucleus of a cell that might be 100 micrometers in diameter, might even be smaller depending on the eukaryotic cell. But obviously the length of the chromosome is much greater than the length of the cell that it's in. So it almost seems necessary, absolutely, that some type of compaction or size reduction of the space it takes to hold this DNA is necessary. So we understand this fundamentally, not totally completely. So I'll tell you what we do know. Now, the term chromatin is one that you'll come across if we haven't come across it already. Chromatin refers to the complex of DNA and chromosomal proteins that are present in the nucleus and, very importantly, during interphase. So if you look at a cell during interphase, the nucleus in general will stain darkly, but you won't be able to see the chromosomes individually. It's very diffuse and it's very spread out, and there are a lot of reasons for this, but one of them is the genes are being expressed. They need to be accessible by various parts of the cellular machinery. So they're not visible at this point. So starting with chromatin, we can say that if we isolate that, which we can certainly do, and do a sort of bulk chemical analysis on it, it turns out that it's roughly 1/3, 1/3, and 1/3, about 1/3 DNA. Now, of course, the DNA is the informational part. I'm almost wanting to say that it's the most important part, but you need all of it. But that's the part that the rest of it is acting to sort of compact. There are special proteins called histone proteins that are very important. They're also conserved and they're in all eukaryotes. And these are basic, and when I mean basic, I mean as opposed to acidic. They're positively charged proteins, and remember that DNA is strongly negatively charged at its surface. So these, for a lot of reasons, but that's one reason that they have an attraction for one another. And then what's left are other proteins that are not histones. These are sort of opposite. They're largely acidic, and there's a lot of variability to them. That's probably the part, those proteins, that we understand the least in terms of their specific role in making a chromosome. But let's think about the DNA and the histones first, because that's really something that's well understood, and it's a fundamental part of the compaction. So histone proteins in eukaryotes have a very intimate connection with DNA. As we just saw, there's roughly, by weight, I guess, there are roughly equivalent amounts of DNA and histone proteins. That certainly implies some kind of stable piece-by-piece interaction between them. And we understand that interaction, so this definitely is the case. So I'm not sure, obviously, if your class or your professor is going to need you to memorize all of these, but it's a good possibility. There are 5 types of histone proteins. They're all named capital H and then a number. So we've got H1, H2A, H2B, H3, and H4. Now, I sort of just alluded to this, but these are really remarkably conserved and similar in all eukaryotes, meaning the amino acid sequence is very much similar, whether it's us, a mouse, even in plants. So this also implies that there's a long evolutionary history between these things, and it implies that there's a fundamental similarity in the way that DNA is packaged in all eukaryotes. Fundamentally, it doesn't mean there are no differences. So let's check out what goes on and relate this to the levels of DNA compaction, because there are generally considered to be 3 fundamental levels of DNA compaction in eukaryotic chromosomes. And the most fundamental and the best understood of these is that association of DNA with those histone proteins. So I'll show you a picture in a moment, which I think will make it a lot clearer. But think of the double helix. So they say that it spools around an octamer of histone proteins. So in other words, there are 8 histone proteins that are arranged in a relatively symmetrical way, forming almost like, I think of this like a spool of thread. The spool being the histone proteins and the thread being the DNA that's going around it. So the octamer, 8 histone proteins, the double helix winds around that, and that forms a fundamental unit of compaction called the nucleosome. Okay? Now, you can actually see nucleosomes under the electron microscope, and it shows nucleosomes as beads separated by short linker DNA pieces. In other words, you wrap it around one spool, then there's a little space, and then you get the next spool. So this represents the first level of compaction. Let's take a little bit of a closer look at that. Specifically, the core histones, as they're called, these are the ones that go into the spool. Most of them, except H1, H2A, 2B, H3, and H4, each of those is present in 2 copies each, and therefore you get the core octamer. In other words, there are 8 proteins altogether in there. The double-stranded DNA gets wrapped around that core in a very specific way, and about 2 complete turns of the DNA can fit around that octamer. This represents about 166 base pairs. Again, I don't know if you need to know that exact number. And then the entire structure is stabilized by the binding of 1 molecule of H1. H1 is the stabilizer that sort of gets this whole thing to stay together in a reasonable fashion to the outside of the structure. So a string of these nucleosomes, and you can actually measure it, is about 10 nanometers in width and creates what's called the 10 nanometer chromatin fiber. And it's hard to prove, but this may represent the way that chromatin exists during interphase. So in other words, even during interphase, chromatin is all spread out. It's still complexed with histones. It's not literally totally spread out. There's some level of compaction there, so it can fit in the cell. Basically, here we have H2B, H2A, H3, and H4, and you can't see it on the other side, but there's 2 copies of each of those next to each other. So this whole thing that looks like a ball here, that's the octamer, okay? This thing is the octamer. And you see the DNA coming in blue here. I'm going to draw my pen in red, but the DNA is going around and back around again, but there's 2 turns going around the octamer, and then this little piece here, that's the linker. You need to have a little space before you can do the wrapping again, otherwise it'll get too tight. And then you see H1 comes along, and you can think of it as almost sealing, like a staple almost going on there and keeping the nucleosome intact and tight. So this shows 2 of them in a row, but you can think of literally hundreds of them going in a row, one going there, one going back down this way. And then here is a transmission electron microscope picture of the 10 nanometer fiber. So I'm not sure if you can see it that well right here, but it definitely looks like beads on a string. It's going all around, and you can see those little linker regions. So the diameter of the whole fiber, meaning the part pretty much from here to here that has the histone in it, is 10 nanometers. Pretty small, but reasonably measurable. I forget exactly. DNA itself, I don't want to say the wrong thing, but it's at least 1/5 of that if you just see a piece of DNA on its own under the electron microscope. So this is the fundamental structure of a nucleosome, and we think the fundamental way that DNA is compacted almost all the time, perhaps. Now, structures have also been observed under the electron microscope, and they're a little bit wider, and the next level of compaction is referred to as the 30 nanometer chromatin fiber. Some people call this the solenoid. So we can see, and I'll show you a picture again in a minute, under the electron microscope, we can see these strings, if you will, with a 30 nanometer diameter present in isolated chromosomes. Our current understanding, which implies that it's really not that perfect, but it makes sense that strings of nucleosomes, right, that those 10 nanometer fibers that we just saw, that they helically sort of become arranged into this 30 nanometer chromatin fiber or solenoid. So there's definite proof that the formation of the 30 nanometer fiber is dependent on H1. If H1 is removed, this will not be able to happen. And we say that this represents the second level of compaction, and the 30 nanometer fiber, this is where we don't really know. It may also represent the way chromatin exists during interphase, but there may not just be one way, and probably there isn't, that the chromosome exists during interphase as chromatin, because When genes are very active, they probably have to be more available. When they're not so active, they can be a little bit more compacted. So the precise arrangement may be a combination of 10 nanometer, 30 nanometer, maybe even some areas that are less compacted and unwound off of the spool. But this picture it is excellent. So we see over here, if you just look at it, you wouldn't probably be able to tell. But it doesn't look quite as much just like beads on a string, and it is 30 nanometers in diameter. So this excellent artist depiction, and I'm just saying it because I certainly couldn't draw that, it looks, so you can see, I don't know if I'm going to be able to draw it, but think of it as a helical structure where that 10 nanometer structure is now going around like this, forming a stable from here to here, 30 nanometers from side to side. And you can see certainly that this would result in much more of compaction going on, right? So also over here, you can see the single nucleosomes. And then, so this fiber, if it was just like that, would be 10 nanometers. And this way, they're going, I can't go back around, right? So it's coming around like this, making a helix, because we can't see the other side. So that's the 30 nanometer fiber, excuse me. The third level of compaction is definitely the least understood. I say here, clearly more compaction is necessary. Remember, we're trying to fit meters of stuff into a tiny cell. So we really need, and by the way, the metaphase chromosome, that's the only time we can really see it in that way that we're used to it. I cannot draw very well, as you've come to understand, but that basically looks like an X, and it's very, obvious under a light microscope. So there's massive compaction that has to occur to get to that. Those other two are very important that we just discussed, but that is not what's going on when chromosomes have to move. When it's time for cell division, the chromosomes say, all right, listen, pack everything up, basically, literally, right? We're going to take a break from gene expression to a large degree. We're going to just pack everything up, and we want to move it. Then when we get to our new home in the new cell, we can loosen things up quite a bit. So the metaphase chromosome, that's the metaphase chromosome, which I've crossed out and then highlighted, are the ones that we see whenever you say, here's a karyotype. They always use metaphase chromosomes. They're the biggest, most robust, most obvious structures there are. So what we believe is happening here is that this is where the non-histone proteins in particular come in and form a scaffold to which the 30 nanometer fiber is then attached, but not in this helical way that I was just implying. Because the evidence suggests the fiber is attached to the scaffold as independent, supercoiled, looped domains. Let's see what that means. So this may or may not be exactly what's going on, but I think you'll get the idea. So right here, each of those is one of those 30 nanometer fibers. And you're looking at this thing head on, but think of it also looking at it from the side. So think of the level of compaction here. Here are a couple of different types of non-histone scaffolding proteins. So when we said that these are independent domains, it means even though it's all one big long line, it's being arranged so that that is going to take up a lot less room than if it was hanging around out. It's attached almost at the same point here, and then another, and then another. And even though you can't see it in this picture, even though they're not technically circles, circles can become supercoiled, right? Which means if you start twisting from one end, they twist up and they take up a lot less space. That's how bacteria do their whole compaction. So each of these independent loop domains can become supercoiled up, which really saves a lot more space. And then here's another scaffold protein. You can see we separated them so you could see. But lots of these, they almost look like flowers or something. Going along in the line, this is probably what's going on. And sometimes when you take a look at a picture, a high quality electron microscope picture of a metaphase chromosome, I draw it like this, but if you really look up at it, you'll see little, almost look like hairs coming out, but these are the pieces of the loop, the 30 nanometer solenoid, all right? So this is the part we know the least about, but this can explain the most compaction that's probably not taking place when the genes are being expressed, but when they really have to get their act together to move during metaphase. So in summary, this is a great picture, I think, and there's a good picture, real electron microscope picture down at the bottom. So you can see just the DNA double helix, right, being wrapped around histones to create the 10 nanometer fiber, then that being wrapped into a helical configuration to make the 30 nanometer fiber, and then each of these being 30 nanometer fibers independently linked to the non-histone scaffold, supercoiled most likely, and lots of them being put along this other non-histone protein scaffolding. So they say chromatin loops here. I mean, when the chromosome is very compacted, we don't call it chromatin, but part of the reason is because it's DNA plus more protein than DNA, right? So what we tend to draw like this, I don't know if you can see it from your perspective, but here's a literal picture of the chromosome sort of lying flat. And if you take a really close look, you'll see all sorts of little spindly looking things, which we believe are the 30 nanometer fiber. And this is an amazing, amazing amount of compaction that's going on here. So this is about as compact as a chromosome can get, but that's okay because this thing, although it's visible under a microscope, is fitting very easily inside of the cell, right? Inside of the nucleus, all 46 of your chromosomes, for example. Well, I should say, let's say it a different way, because I could hear some of you saying already, and I said it to myself. No matter what, all the genetic material is fitting inside the nucleus, right? But even with the 10 and 30 nanometer compaction, it's still very diffuse. So my point is, when these chromosomes get together, and you can literally say, there's number 1, there's number 2, there's number 3, they're very tightly wound, and they are, you can see right in that one chromosome, you can imagine how long that piece of DNA is, and yet there it is. In fact, I know I used the spool analogy before, but this is like a mega spool. You think of all the DNA wrapped around these scaffolds in a way, because it's really easier to carry this way, really. When cells have to divide, you want to grab one whole chromosome and take it. You don't want pieces of DNA falling out and sitting around. You want an easy to transport type of a structure, and that's exactly what this is. So the bottom line is that obviously there has to be some way that the DNA that we all know and love is compacted up, ultimately perhaps into that metaphase chromosome, but there are a couple of levels in between that have to do with the way the DNA exists, even as chromatin in the chromatin during interphase. So hopefully we'll be learning more, and I mean we as a scientific community will be learning more about this in the future, but for now, that's the story about chromosome compaction. I'll see you soon.